Hey guys, welcome back to the Func Med Nation podcast. It is Dr. Steve Noseworthy here. And today we've got a great podcast interview with researcher and scientist Dr. Nathan Bryan. We're talking about nitric oxide. This is actually going to end up being a two-part interview. But I want to let you know before we get into that I took the summer off. We did a lot of traveling, spent a lot of time with family. And so you may have noticed that I've been silent for the past 4 months or so. And so I'm back into the swing of podcasting again. So you'll see these podcasts and YouTube videos coming back up online again. In fact, I published one, I think, just today with an interview I did not long ago, Dr. Jason Richardson. So you can look for that. By the time you see this one, you may have already listened to that podcast or watched this YouTube video. But I wanted to let you know that the interview with Dr. Nathan Bryan, it's actually really good. I went back and listened to it. I had to edit it. We did have some issues with the internet connection. And so I did my best to edit out the glitches. And so you might notice that the audio can be just a little bit rough at some places, but you're going to get the depth of the information that Dr. Bryan is sharing as we go through the conversation. But it ends somewhat abruptly because we spent pretty much an hour and a quarter with some pretty decent connectivity and flowing conversation. But then all of a sudden we started having issues, but we had invested too much time to just scrap it and start over. So we ended it abruptly, as edited out the last piece of that, and just trust that we will resume in another episode. And we'll go back and we'll finish the conversation that we started because we weren't done. I had too many more questions and I couldn't let them off that easy. So enjoy, and thanks for being a listener, and we'll talk to you again pretty soon. And here's the interview with Dr. Nathan Bryan. Hey guys, welcome back to the Func Med Nation podcast. I'm your host, Dr. Steve Noseworthy. I have a tremendous pleasure of talking today with Dr. Nathan Bryan. Nathan, you probably don't remember this. You and I have actually spoken before, back when you first started NeoGenis. We had a very brief conversation. I can't even remember what the context was, but I'm talking probably what was that 2012 that you started that company?
I started that company in 2009. We brought product to market in 2010.
Okay, so it was probably somewhere around there because you had been on the market for just a little bit, and I had a clinical question. I was not completely new to functional medicine at that point. I had a few years under my belt, and I was teaching functional medicine, and I had a question about nitric oxide and all that stuff, and you were so kind to spend a few minutes on the phone with me just to sort out whatever it was that was going through my head. I kind of jumped into the flow. So why don't you take a couple of minutes and talk about your background so that the people who are listening, who are predominantly functional medicine practitioners of different types, so that they kind of have an idea of where you've come from. Always nice to know that the expert is the expert, which you certainly are. So why don't you take a minute and talk about your background, and then we can get into the clinical stuff.
Yep. Well, thanks, Dr. Noseworthy. It's great to be with you, and always appreciate the time. And to really tell the story about nitric oxide, I think it's really an important molecule that not only far too few clinicians know about, but the patients and consumers in general. But, I grew up in small town Texas, boy. I've been independent for many, many years. I think when I was 15, I moved out, had my own apartment. So I've been kind of doing my own things since an early age. But, I graduated high school and went to the University of Texas at Austin. I got a degree in biochemistry. Quickly realized there wasn't a whole lot to do with a bachelor's degree in biochemistry in terms of the job market. I went out and worked as an analytical chemist in phenol and chemical for a couple of years, and it was just too monotonous and really wasn't stimulating at all. So I quickly realized that I probably needed to further my education. I then enrolled at LSU School of Medicine, I think it was 1999 or 2000. I'd taken a 4-year sabbatical to kind of find my way and figure out what I wanted to be when I grew up.
Yeah.
Then that's when I got introduced to this whole science of nitric oxide. I was doing my PhD in molecular and cellular physiology, and I was working in a lab of Martin Felix, who was a pharmacologist from Germany. He'd been in nitric oxide field since probably the early 80s. Was vice president of a pharmaceutical company that was trying to bring to market safe and effective nitric oxide therapeutics. Well, after LSU, when I completed my PhD in 2 years, I then went and trained in Boston at Boston Medical Center.
Yep.
I was doing a fellowship at the Whitaker Cardiovascular Institute and still continued the research on nitric oxide, particularly in vascular biology. And by that time, I think I'd published a couple dozen papers as a student and as a postdoc. And I was then trying to figure out, how do I get back to Texas? Boston was a culture shock for me.
Sure.
I enjoyed my time there, but was really trying to figure out, how do I get back south? And I contacted Fred Murad, who had just won the Nobel Prize for the discovery of nitric oxide, and he invited me to come and give a lecture at the University of Texas Medical School in Houston for a faculty position. And I flew down, gave a lecture, and he offered me a position. So I think I started at UT as an assistant professor of molecular medicine in maybe 05 or 06, and then spent, 8 or 10 years there as a professor of molecular medicine, and really just trying to advance the science of nitric oxide. I had a lot of kind of patent ideas, kind of innovative ideas when I was in Boston, but it really wasn't encouraged to file patents and to innovate. So the first thing I did when I got to UT was I just enabled patents. I did experiments, I did invention disclosures. We then submitted patents, and over the next 3 to 4 years, patents started being issued. And because I knew early on that nitric oxide had the potential to change the world. If we could figure out how to make nitric oxide in a safe and effective kind of product technology or even a drug, then it would change the face of healthcare and really get to the root cause of many so poorly managed chronic diseases. And so that was my compass. I had my blinders on for a number of years, and that's all I did, and that's all I've done now for the past 25 years of my life.
It's interesting you say you put the blinders on, and I know why you phrase it that way, but one of the things that I've noticed, not just talking to practitioners, but also researchers in different disciplines and areas of expertise, is that quite often they intentionally blind themselves to the larger picture of human physiology because they're so dedicated and focused, either because it's a topic of interest or because maybe there's some, maybe business considerations, like forget everything else, focus on this. Do you feel like with your career path and your studies, you've obviously focused quite a lot on nitric oxide, but because nitric oxide is pretty much ubiquitous and it's linked into so many different systems, do you feel like you've got a good grasp of the totality of human function? Or do you feel like your research has really kind of siloed you in your knowledge and expertise? And I certainly don't mean to offend you by asking the question. I'm genuinely interested.
Yeah, no, it's a good question. It's a fair question. It's one of the reasons I left academia, because for me, there's over 185,000 scientific publications on nitric oxide.
Wow.
We know basically everything about the enzymology, biochemistry, and physiology around nitric oxide. The challenge in the field is, how do you restore endogenous nitric oxide production and correct the dysfunctions in human biology?
Yeah.
So that's really what changed my focus. So as you mentioned a very good point, nitric oxide is ubiquitous in every biological system, and there's not a single disease where nitric oxide is not involved. Because the hallmarks of every single chronic disease, and it doesn't matter if it's cardiovascular disease, Alzheimer's, diabetes, autoimmune disease, the hallmarks of that disease. It's low blood flow, which we call ischemia.
Yep.
There's inflammation, oxidative stress, and immune dysfunction. And nitric oxide is what controls and regulates every single one of them. It vasodilates, so it restores blood flow to that dysfunctional tissue or organ. It mitigates inflammation, it reduces oxidative stress, and it prevents the immune dysfunction that occurs in that tissue. So it has siloed me, but I think this is considered the holy grail in cardiovascular medicine, nitric oxide. And there's a vascular component to every single major chronic disease. So we can solve basically the mechanism of any major chronic disease simply by understanding how nitric oxide works, how it's produced in the human body, what goes wrong in people that can't make it, and then how do we fix that?
Sure.
So you're right. I have had blinders on because I knew that was a big enough bite for me to take. They would keep me busy for my entire career. I didn't need to focus on anything else.
So one of the questions that comes to my mind, particularly with your background in drug discovery, which was a phrase I wasn't familiar with, and if you give me a second, I'd like you to expound on that. What was it that made you go from what you were learning about nitric oxide and its role in health and disease, and go down the nutraceutical pathway as opposed to the pharmacological one?
Look, it's a very good question, and it's circumstance, right?
Hmm.
And it's just acute observations or astute observations. So I'm trained as a drug discovery chemist. My objective in getting a degree in biochemistry and physiology was, and long term, even I knew this in high school, that we could understand the mechanism of disease to the extent that we could then restore and repair the function of that tissue or disease process.
Right.
So there's several ways to do that. I mean, and what I've learned over the past 25 years is chronic disease is caused by two things and two things only. The body's missing something that it needs, or it's exposed to something toxic. And so in that paradigm, there's really no room for drug therapy. Drug therapy as we currently know it, and that big pharma knows.
Right.
So if you can figure out what the body's missing, 95% of Americans are deficient in iodine, 85% of Americans are deficient in magnesium. Another large population is deficient in selenium, chromium, all these other trace minerals and nutrients. And those are necessary cofactors for a lot of biochemical reactions.
Right.
So that's one aspect. And you can so dietary supplements work great affecting that particular portion of the population. Then the other aspect is if you figure out what's toxin you're exposed to, whether it's latent infections, root canals, exposure to environmental toxicants, herbicides, pesticides, EMF, and then rid your body of that.
Yeah.
The human body is regenerative by nature, so the body heals itself. So once we figured out that nitric oxide can be affected, the nitric oxide production is affected by not just dietary nutrients, such as nitrate and nitrite from green leafy vegetables, but also some antioxidants and what we call redox couplers to restore the function of the NOS enzyme, then we could do that through a dietary intervention. And the other thing, this was, not encouraged by my superiors at the University of Texas, because when we first intimated that we were going to bring to market a dietary supplement, I had zero support from my superiors, because we're in the business of drug discovery, diagnostics, medical devices, drug therapy that can treat, cure, prevent, or mitigate human disease. And dietary supplements aren't intended to do that by nature.
Right.
And what we did was, and what they not just recommended and suggested, but almost forced us to, was to do randomized placebo-controlled clinical trials. Go through the same rigor and regimen as you would in a drug study. And we did that with our dietary supplement, and it works great. There's no other nitric oxide product on the market that does what our nitric oxide technology does. In fact, it's so safe and so effective that we now have a drug company where we have nitric oxide drugs in phase 3 clinical trials.
Wow. So is that going to supplant the nutraceutical side, or do you think that there's room for both?
Well, I think there's a both. Just like, there's prescription fish oils for hyperlipidemia.
Yeah.
And there's over-the-counter fish oils for, dietary supplement. Obviously not for treating hyperlipidemia, but it's dose dependent, right? So it's similar to over-the-counter Tylenol. Or if you go to the hospital and you're in severe pain, they can prescribe you a higher strength Tylenol or some NSAID. Right. So these types of paradigms have been set before, and there's precedence for what we're doing. So we can take our core nitric oxide technology, put it in a dose that falls within the range of so-called normal dietary exposures, and make it a dietary supplement. And there's a range where it has to be administered under the physician, that we can file investigative new drug applications, go through FDA-approved clinical trials, and then have a product or a drug that's under the supervision of a physician that can write a prescription for that particular drug. So there's room for both of those, and it's dose dependent.
So does this have, and I believe it does, nitric oxide has a hormetic response curve, right? Too little is bad and too much is bad as well. Do we have any idea what the sweet spot is, and how do you divide that between, this population fits on this part of the curve and a nutraceutical approach is okay for them versus someone's beyond that threshold and we want to go to the pharmaceutical higher dose dependency.
Yep. No, look, it's just like any molecule. There's a U-shaped curve where we know too little is bad, too much is bad. So you gotta find that sweet spot. Fortunately, we recognized that early on, because the toxicity of nitric oxide has been defined for many, many decades. There's only two causes of toxicity. It's methemoglobinemia, which means that you're exposed to too much and it oxidizes the iron of red blood cells and reduces the oxygen carrying capacity of the red blood cell, and you become cyanotic. You'll get blue around the lips, and you basically suffocate. And that's massive doses. And the other sign of toxicity is low blood pressure or hypotension. So if you take too much nitric oxide, you get systemic vasodilation, you lose perfusion pressure, you develop orthostatic hypotension, low blood pressure, hypoperfusion to end organs, and eventually end organ failure. So what we do, and we've monitored this now in many, many different patient populations, from pediatric patients to hypothyroidism patients with low blood pressure to begin with, to the sickest of the sick patients over the past 3 years during the COVID pandemic. And the amount of nitric oxide that we dial in with our drug therapy is extremely safe. We've treated over 600 really sick, at-risk, highly susceptible patients, not a single adverse event, not even a headache. So no safety signal whatsoever. And these are polypharmacy patients, extremely sick, vulnerable from COVID-19, and there's no safety signal whatsoever. So we were very fortunate. Well, I don't say fortunate. We were very deliberate, because we knew exactly what we were doing, how we were doing it, and we need to deliver the right amount of nitric oxide at the right time to the right patient. There's not a single disease process that would not be benefited from that type of therapy.
So is it safe to say that if you're taking a nutraceutical approach to manipulating nitric oxide in some way, that you're really not going to get in trouble with toxicities, that you would have to consume an inordinate amount of lozenges, say, to get to that point?
No, that's exactly right. So if you take these as instructed, then we've never had an issue with toxicity or any adverse events in people. But, it's human nature that, and people think, well, if one lozenge is good, 3 or 4 is going to be great.
Yeah.
And that's not right. So we have to avoid that temptation by human nature. And so, but we recommend, one lozenge 2 to 3 times a day. And it's as you know, as a clinician, you can't ascribe a one size fits all, because everybody's different. Yeah. For me, I just turned 49 several months ago. I'm in good health, normal blood pressure. So I take it kind of as a prophylaxis or as a maintenance. And so I'll take 1 or 2 lozenges a day. I take our beet powder before I go to the gym. And that seems to do me very well. But that's much different than, say, a 70-year-old hypertensive diabetic patient with erectile dysfunction. The demands on him are much different than the demands on me. So obviously that patient's going to need a different type of regimen than, say, me, just for maintenance and trying to prevent getting sick or prevent this age-related loss in nitric oxide.
Right, and we haven't even gotten into the conversation where we actually define what nitric oxide is, how we get it.
Oh, yeah.
And I think we should probably cover that next. But you brought up this idea of kind of a scale of needs, where you as a healthy 49-year-old are taking it from a prophylactic standpoint, but you may have a very complicated clinical scenario where somebody needs to take more than 1 or 2 lozenges. And we'll talk about your products probably towards the end of the discussion. And so do we have any diagnostics that guide decision making? Number one, in terms of identifying people who are nitric oxide deficient or who have some kind of a skew in the nitric oxide synthase enzyme profile, if I can use that phrase. Do we have any diagnostics that will clue us into, hey, this is a problem and it looks like this? And do we have any guides that might lead into prescribing frameworks, decision-making frameworks, or is it just kind of like, this is really bad, so we're going to give you more? Where are we with that?
Well, we've come a long way in 20 years, and I think we're still a long way from where we need to be, but we've made progress. And so as long as we're making progress, I'm happy with that. But, it's not like your cholesterol or vitamin D or triglycerides where you go to your physician, draw labs or blood, and then give you a number.
Right.
And I think it gets to the point of what is nitric oxide. So it's a gas that's produced by the lining of the blood vessels and other cell types. But once it's produced, it's gone in less than a second. So we really can't pick up this gas in blood. So to answer your question, there's no blood labs that we can rely on. Now, there's consequences of insufficient nitric oxide production that show up in perturbations like triglycerides or C-reactive protein or sedimentation rates or ferritin, kind of these markers of inflammation. So let's go back to kind of genetics. If we do genetic screening, so obviously if you have a SNP, what's called a single nucleotide polymorphism in the ENOS gene, then when you go to translate that DNA code into protein, that protein is going to be dysfunctional because there's a problem with the genetic code. So obviously those patients who have a SNP in the ENOS protein are going to be nitric oxide deficient. The other common one is MTHFR, this methyl tetrahydrofolate reductase enzyme. And so people understand the importance of it because it's a default in the methylation cycle. But what they fail to recognize is it's also the enzyme that converts biocrin to tetrahydrobicrin. That's the rate-limiting step in nitric oxide production. So MTHFR patients have a deficiency in nitric oxide production.
Right.
And that's, depending upon which report you read, maybe 45 to 55% of the population in the U.S. So it's a majority of the people. So what we rely on is really, and going back to, that was the first question we tried to answer back in 2009, because we were bringing to market a nitric oxide product, and the first question we encountered was, how do I know if I need this?
Yeah.
A very important question. Right?
Yeah. Yeah.
So even back then, the genetics weren't where they are today. There certainly weren't the diagnostic labs. So I developed a salivary test strip. And now these have been on the market, I guess, for 12 or 13 years. But you just apply some saliva into this test strip, and hopefully it turns bright pink. Then that tells you your body is making nitric oxide from some source, although it doesn't tell us where it's coming from. But if it doesn't turn pink, then it tells us your body's not making nitric oxide. And then we have to interrogate kind of what goes wrong in these individual patients. But so those are kind of the tools you can have in your toolbox. But really, we rely on symptomology. So if you've got a patient that has high blood pressure, they're nitric oxide deficient. If you've got a patient with erectile dysfunction, they're nitric oxide deficient. If they become exercise intolerant or short of breath by walking short distances or flight of steps, they're nitric oxide deficient. If they have mild cognitive disorders, even vascular dementia, those are symptoms of nitric oxide deficiency.
So I have, to be honest, I have like 800 questions, which is one by one. Which is like, anyone listening to the podcast have heard me say that so many different times. But, I start thinking about different clinical presentations, right? It's kind of a no-brainer. It's really black and white. Nitric oxide causes vasodilation. So if you're hypertensive, you probably have an issue with that. What about other clinical scenarios where we might see, let me just kind of play this out, right? We look at the three different nitric oxide synthase isomers, and you've got endothelial, neuronal, and then immune or inflammatory, depending on how you want to translate the I. And my understanding is nitric oxide is nitric oxide is nitric oxide. Doesn't matter what tissue or which enzyme pathway is making it. But you can have upregulation or downregulation of INOS as compared to, say, endothelial. And you may have somebody who is with, let's say that they're chronically, we call it functionally hypotensive, and we tend to define that as anything less than 110/70. So let's just say they've got low blood pressure, but at the same time, they have some kind of an autoimmune issue where theoretically their INOS pathway is upregulated, making much more nitric oxide than those two other, let's call it more favorable pathways. Is it possible that we might get false signals in the sense that a salivary test will show a lot of nitric oxide production, and that actually is part of the problem if the context is someone with autoimmunity who's locked into an inflammatory cycle? Is that within the realm of possibility?
Yes, it's certainly within the realm of possibility. But what we're measuring with that salivary test strip is really the presence of an oral microbiome that reduces nitrate the primary oxidation product of nitric oxide into nitrite in the oral cavity. So here's the issue. You can have good endothelial function in your body making sufficient nitric oxide in the lining of the blood vessels, your normal tensive. You eat a good diet, but if you use mouthwash, if you have an oral infection or oral dysbiosis, you can't convert that nitrate into nitrite. And still, you may have good endothelial function, but your global nitric oxide production is becoming compromised. So there's two pathways, and one can compensate for the other. But when you start to lose the ability to produce nitric oxide from both, that's when the symptoms start to set in. You know, we published on this, I believe it was in 2004, and you bring up a very good point, this iNOS, or inducible nitric oxide synthase that occurs under an acute or chronic inflammatory condition. We published on that. So when you have chronic overexpression of iNOS, it leads to feedback inhibition of the constitutive isoforms of NOS.
Right.
So these people, whether it's, you know, rheumatoid disease or inflammatory bowel disease, or no matter where the acute or the chronic inflammatory response is, those patients become systemically and globally nitric oxide deficient. But there's also, you know, nitric oxide, you made the comment, nitric oxide is nitric oxide is nitric oxide. It is, but it also, the metabolic fate of nitric oxide is dictated by the environment in which it's produced. So if you've got a chronic inflamed condition, let's just take inflammatory bowel disease, because we've published on that a number of times. In that inflamed gut, you've got upregulation of NADPH oxidase, myeloperoxidase, all these oxidative enzymes. And that iNOS enzyme is producing a lot of nitric oxide.
Right.
And then the reaction of nitric oxide with superoxide is kind of rate limited. It's a diffusion limited reaction. So it's very fast. The reaction of nitric oxide with oxygen is second order with relation to nitric oxide. So you need a lot of nitric oxide to react with oxygen. That forms nitrogen dioxide and all these higher nitrosative end products that are, you know, that can nitrate proteins or nitrate fatty acids and lipids and cause cellular damage. So as long as it's kind of slow and constitutive production of nitric oxide, that shuts down superoxide production from mitochondria, from NADPH oxidase, and even from eNOS. So it's self-regulating. But in a chronically inflamed person, you've lost regulation of that.
Right.
So what you have to do is figure out what's the insult they're exposed to in the environment. And if that's the case, then you got to remove that source of exposure. But then, and it's not really counterintuitive or paradoxical, that you have to restore the constitutive production of nitric oxide. And that downregulates the iNOS and all the oxidative stress that's occurring in that inflamed tissue.
So is it implicit in that statement then? Is it true that if you do things to upregulate eNOS and nNOS, like endothelial neuronal, that you'll automatically downregulate the inducible form?
I'm not sure if we can confidently say that. We know the opposite's true.
Yes.
That if you downregulate iNOS, you can kind of lead to more constitutive production of eNOS and nNOS. If we restore eNOS and nNOS function, not if you don't remove the cause of the inflammation. So if you've got a person that's eating, you know, if they have a dairy or gluten sensitivity and they continue to expose themselves,
right,
then they're going to get that inflammatory response every time they're exposed to that toxin. And that's automatically going to upregulate within 4 hours of exposure an increase in expression and activity of iNOS. So that's just the innate immune response on the inflammatory cascade or for wound healing.
Right.
That's just the body doesn't discriminate. So you've got to figure out what's causing the inflammation and then get rid of that for the patient.
So, I'm, you know, as we have these conversations, I'm always trying to translate that into something I can do better or different clinically. Does that mean then, you know, as much as we might want to say absolutely everybody should be on, let's say your nitric oxide products from day one, is it more of a case where clinicians have to be really judicious and say, well, step one might not best be upregulating nitric oxide production. Maybe we have to work on the inflammatory state and triggers first and then bring that in as, let's call it a tier 2 strategy, but a necessary tier 2 strategy. Would you agree with that based on your knowledge of the physiology and the research?
But when you have inflammation, part of the inflammatory response is an increase in oxidative stress and the consequential immune dysfunction.
Yeah.
And that's what's causing the tissue and cellular damage. So we know nitric oxide prevents oxidative stress.
Yes.
And we know nitric oxide prevents the upregulation of adhesion molecules on the endothelium and prevents cellular migration and adhesion to the vascular endothelium. So I would think that nitric oxide should be foundational, kind of the first step in the process of any chronically inflamed person, because you have to calm the inflammation. You have to put out the fires of inflammation. And nitric oxide is the best thing I've encountered in my 25-year scientific career that does that at the cellular and subcellular level.
Right.
So whether you have uncoupled mitochondria generating superoxide or an overexpression of NADH oxidase, nitric oxide recouples the electron transport chain. It shuts down NADH oxidase production of superoxide, and it recouples the NOS enzyme. So we're eliminating oxidative stress. And then if you've got vascular inflammation, which occurs in every single chronic disease, then it downregulates the adhesion molecules. It prevents monocytes and neutrophils from sticking to the lining of the blood vessel, extravasating through and causing the immune dysfunction that's characteristic of every chronic disease. So I think, and what the clinical evidence, both the scientific and clinical evidence provide, is that nitric oxide should probably be the first step in treating chronic inflammation until you can figure out and interrogate the individual patient. What are you exposed to that's causing the inflammation?
Yeah.
But in the meantime, we're gonna put out the fire.
Put out the fire. Yeah. So it makes me think that because one of the things that I practice and I've taught in seminars over the years is that if you're going to play around with the nitric oxide system, first and foremost, try to figure out if you've got some dysregulation of the nitric oxide synthase enzymatic profile, and work on that before you do anything like giving arginine, for example, as a precursor.
Oh, yeah.
And I think we can use this to circle back around and then talk about nitrites and nitrates and nitrosamines and try to sort all that out. But, my understanding is that your approach in terms of having a lozenge with nutrient-based ingredients that works with the oral microbiome to increase nitric oxide, which then can go on and do everything that you just said, is completely different than taking a patient who has an upregulated iNOS pathway and downregulated endothelial neuronal and taking arginine and dumping a whole bunch of raw material into that person, which theoretically would flow through the INOS pathway and just upregulate a massive amount of production of downstream metabolites that are things like superoxide radicals, especially if you've got someone who has problems with MTHFR or the THB production and all of that kind of stuff. Is that, I just want to make sure that my thought process is on the right track with that.
No, you're exactly right. So, arginine is a semi-essential amino acid, meaning that we get it from the breakdowns of proteins we eat, whether they're plant-based or animal-based proteins, but it's also generated through the urea cycle, the partial urea cycle, which is part of every cell in the human body. So we make enough arginine through the urea cycle to fully saturate the binding site of the nitric oxide synthase enzyme. So there's never a condition of arginine deficiency, except one. That's a rare condition called arginosuccinic acid urea, or ASA. So giving more L-arginine actually causes more harm than good for the reasons you described of, feeding and fueling that iNOS pathway. Yeah, so the human body is self-regulating by nature. So if the human body sees too much of something, it tries to get rid of it or detoxify.
Yep.
And it's no different with arginine. So when we give high dose arginine, the body responds by upregulating arginase 2, which is an enzyme that diverts arginine away from producing nitric oxide, and then it's excreted as ornithine and urea, because you can get into nitrogen imbalance. And so that's the body's self-regulating pathway to prevent hyperammonemia and nitrogen imbalance.
Yeah. So
I think arginine has never been the answer in terms of nitric oxide production. There's been a lot of misinformation and confusion in the marketplace because those products came to market, I think, in the late 90s, shortly after the Nobel Prize was awarded, when it was realized that arginine can be used to make nitric oxide. So let's put arginine in everything and call it a nitric oxide product.
Yeah.
Yeah, and I think that's the problem with these companies who market this. Either they don't understand the biochemistry in these products, or worse yet, there are companies out there who understand it, but are deliberately practicing deceptive trade practice and outright fraud in selling these products, which they know have no benefit to the human, and in fact could cause more harm than benefit.
Yeah. So how do you interpret? In fact, I can't remember where I was, but somewhere just recently, I had another doctor tell me, he personally, he's a little bit older than I am. And he said something like, I take arginine and citrulline every night before I go to bed, and it's wonderful. My hands aren't cold. I wake up with a morning erection all the time at my age. And he was trying to relate this to nitric oxide. So what do you think is going on for people who do take arginine or citrulline and get some kind of a clinical benefit? Is it just a different pathway, different mechanism? Or is there yet more to be discovered about nitric oxide?
Well, I don't think, perhaps there's something more to be discovered around nitric oxide, but not as it relates to the 5-electron oxidation of L-arginine to nitric oxide. That pathway is very well elucidated from the transfer of those 5 electrons in a multi-step oxidation reaction. Arginine has been shown to increase human growth hormone production. There's lots of metabolic activities or metabolic paths for arginine. Nitric oxide is just one of them.
Gotcha.
Right? Citrulline is a byproduct of nitric oxide production. Then citrulline gets caught up in this partial urea cycle, and it's converted into arginosuccinic acid, and then arginosuccinic acid is converted back to L-arginine. So citrulline is a long way from nitric oxide. In fact, citrulline is a byproduct of nitric oxide production. And in most biochemical reactions, there's a feedback inhibition on that. So citrulline, especially at high doses, I tell people don't waste your money. But here's what also I tell people: look, if you're taking something that you're getting benefit from, then don't get away from what works. They're known placebos. That's why you do placebo-controlled clinical trials, because people experience a placebo effect. The mind is a very powerful organ. So if people are getting a benefit or thinking they're getting a benefit from a product that contains arginine or citrulline, then by all means, keep taking it.
Yeah.
But just be careful in the fact that you're not doing more harm than called unanticipated side regulating your nitric oxide production as a consequence.
Yeah. Yeah. And I tell people all the time, because they'll say, hey, should I keep taking this? They might send me like a picture of a supplement. And my next question is, well, is it helping you in some way? Because I'm fully open to the fact that I certainly don't know everything. And I'm like you, if it helps, and if you think reasonably you're not doing any harm, I really don't think there's any problem with that. Why don't we back up? Because I realize I keep saying we're going to do it, but let's talk about dietary influence over nitric oxide production. I'd like to revisit this concept of the oral microbiome and ask whether or not the gut microbiome plays a similar role. But let's talk about nitrates, nitrites in the diet, the difference between that and nitrosamines. I have to tell you this. This came to mind when you agreed to be on the podcast with me. I said, I've got to tell him this. So back when I was in my undergraduate, we're talking back in the 1980s, I played ball with a guy who was doing basic sciences, and I think he was doing a biology or chemistry course. And we were playing outdoor volleyball, and we were barbecuing and all this kind of, and he was eating hot dogs, and everyone was going, oh, you can't eat hot dogs because of the nitrites. And he said, well, and he's like, I just learned this in chemistry class, that if you drink orange juice at the same time you eat a hot dog, then it kind of neutralizes the nitrites. Now, this is, again, back in the 1980s, and we now know a lot more vitamin C and how all this kind of fits in. I thought that was a hilarious. It just popped right into my mind as I was going through some of your research papers. But let's back up. Let's talk about dietary nitrites, nitrates, the relationship to nitrosamines, what we have to be aware of and careful of. And then once we eat something that has the potential to drive nitric oxide production, what's going on?
Yep. Now, look, it's a very good question. In fact, one I've been dealing with for more than 20 years, because, it's maybe difficult for you and your audience to believe, but the media doesn't always do a good job of telling us the truth. Right?
Say it's not so.
This goes back to the 1950s. And, most scientific discoveries are made just on simple observations.
Yeah.
Right? So and we call this nutritional epidemiology as it relates to nutrition. So you can see a trend or an increase in certain types of cancers by people who eat a certain amount of cured and processed meats. So I never argue with the data. It's the interpretation of the data that's subject to argument. So in order for nutritional epidemiology to make sense, you have to have a biologically plausible mechanism for that interesting observation. So back in the 60s, they come up with this explanation that the nitrite forms nitrosamines, and then secondary low molecular weight nitrosamines can intercalate and mutate DNA and cause cancer. That was their biologically plausible mechanism. And then in the 1980s, it was discovered that the human body produces nitrate and nitrite. Those are the first observations that eventually led to the discovery of nitric oxide. So then why would the body naturally produce a carcinogen, right? And if their biologically plausible mechanism was true, then vegetarians would have about a 10-time higher rate of cancers than meat eaters, because 85% of the nitrate and nitrite we get from our diet comes from green leafy vegetables.
Right.
Only 5% comes from cured and processed meats. So now we know that entire kind of story has fallen apart. And we know that the protective benefits of a plant-based diet or a vegetable-based diet is because of the nitrate content and because it goes to generate nitric oxide. So that's the divergence. But we still have to worry about nitrosamines. I mean, nitrosamines are known human carcinogens, a certain class of them. So what we have to do is figure out, okay, primary, tertiary, and quaternary amines aren't affected, because primary amines, if they're nitrates, they just rearrange to form alcohols. Tertiary and quaternary amines, there's too much steric hindrance and you never form a nitrosamine. So it's only the secondary amines that we worry about. Low molecular weight, things like dimethylamine. If you form N-nitrosyl dimethylamine, NMDA, that's a known human carcinogen. So the chemistry's been well described for 50, 60 years. And the Code of Federal Regulation changed, I believe, in 1973 by the U.S. government, that they required ascorbic acid or erythrobate now to be added to any cured meat product where nitrite's added, because that's known to inhibit any nitrosated chemistry. So for that to happen, you have to have a low molecular weight secondary amine. You have to have the nitrosated chemistry happen, and then that nitrosamine has to be ingested, intercalate DNA, cause mutations, and cause cancer. But things like vitamin C, vitamin E, any antioxidant will prevent that nitrosated chemistry, even if there's a secondary amine present. So what we do is we don't have any secondary amines present in any of our product. And even if we do, or even if these people have eaten something, we provide super stoichiometric amounts of vitamin C to effectively inhibit any potential nitrosated chemistry from occurring. So we don't worry about nitrosated chemistry at all in the methods that we develop, and that's why we develop the lozenge, because we can control and dictate the metabolic state of nitric oxide. That's why we develop powders that you put in water. We know when we control the solution chemistry, so when you consume it, we know what we're doing, and there's no side reaction going on.
So the phrase you use, the phrase super stoichiometric vitamin C, does that refer to enough vitamin C to prevent this from happening?
That's exactly right.
Yeah. So what you're saying ultimately is that I need to contact my friend Don and apologize for laughing at him back in 1984.
He was before his time. He was wise beyond his years.
That's really funny. All right. Yeah. So I've read some of your papers where you identify, I believe it's 4 keystone bacteria that live in the crypts of the tongue that convert dietary nitrites into nitric oxide. I have other questions, but let's just kind of focus on that. And if we can, are there any takeaways from your research or your understanding of other research that has any implications for things like oral hygiene? I know you've published papers on, hey, don't use mouthwash if you want nitric oxide to help you. So what can we take away clinically from all that, and how does this work?
Well, we started investigating this more than probably right at 20 years ago. The Human Microbiome Project was in full swing, trying to map out the microbiome, and most scientists and researchers focused on the gut microbiome, a critically important part of the human biology. As you know, the bacteria that reside in and on our body outnumber on human cells 10 to 1. So there are 10 times more bacteria that form us as humans than our own human cells.
Yeah.
And anything we do to disrupt that microbiome causes human disease. right? And so there's a reason we don't take antibiotics every day for the rest of our life, because of the known havoc it causes on the microbiome and human disease. So we started interrogating the oral microbiome, because nobody had really focused on that. There was a couple of papers published, I think, in the early 90s. And so we started trying to figure out, okay, can we culture certain bacteria that have nitrate or nitrite reductase activity that can provide a source of nitric oxide to the human host? And does this have any vasoactive consequence? Meaning, can you correlate this with blood pressure?
Right.
And we published this, I believe, in 2011, where we did tongue scrapings. I think we enrolled, I don't know, maybe 50 or 60 patients in the Texas Medical Center. And we just did tongue scrapings. We cultured the bacteria, and then we correlated it to their blood pressure. And what we found was that the people who had the most diverse oral microbiome, and we could actually culture and identify these nitrate reducers, had normal blood pressure. The people who had a least diverse oral microbiome had the highest blood pressure. In fact, there were no nitrate-reducing bacteria that we could culture from these tongue scrapings. So that was a nice association, but not causation. Again, now we have to take it a step further. So we followed up that study, and we published this in 2015, and we found that if we just enrolled normal, healthy, normal-tensive patients, people with good blood pressure, and then we give them mouthwash for 7 days, twice a day, we eradicate their oral microbiome, their blood pressure goes up, in some cases 20 millimeters of mercury. And then 4 days after we stop the mouthwash, the microbiome is really resilient in that it restores and repopulates. And then after 4 days, we can normalize the blood pressure of the people that we'd caused hypertension. So that was the aha moment. And so this was the first indication that the presence of oral bacteria were regulating systemic blood pressure.
I mean, that's not insignificant.
No, that we have an epidemic of hypertension, right? 2 out of 3 Americans have an unsafe elevation in blood pressure. And yet you go to your primary care doctor, cardiologist, they put you on an ACE inhibitor, an ARB, a beta blocker, or a diuretic, or calcium channel antagonist.
Yeah.
But yet 50% of the people that are given those drugs don't respond with better blood pressure. And what we've published on in our story we're making is their hypertension is a symptom of oral dysbiosis. So they're not going to respond to standard ACE inhibitors, ARBs, calcium channel antagonists, because that's not the cause of their hypertension. We have to restore the oral microbiome and the ecology of those bacteria, and then you restore normal blood pressure. So for us, that's a game changer. I mean, if you can get people off, 2 out of 3 Americans use mouthwash every day. 2 out of 3 Americans have an unsafe elevation in blood pressure. That is not a coincidence. We and others have published that it's causal. So if you're using mouthwash, you have to stop using mouthwash. If you're using fluoride in your toothpaste, you have to give it a fluoride. Yeah, the other consideration is fluoride. You know, fluoride is in the drinking water. It's in most toothpaste.
Yeah.
Fluoride's an antiseptic. That's the reason they put it in the water supply, to kill the bacteria. It's a neurotoxin, and it kills your thyroid function. So, you know, there we have an epidemic of hypothyroidism.
Right.
And fluoride is a major contributor to that. So get rid of your fluoride toothpaste and stop using mouthwash.
What about
that will allow now your oral microbiome to repopulate and do its job.
What about things like tongue scraping practices? What impact does that have?
You know, part of that paper we published in 2014 revealed that people who did tongue scraping once or twice a day had a better blood pressure profile than those who didn't. But to the contrary, if you do tongue scraping and use mouthwash, then the increase in blood pressure from the mouthwash is the highest we've seen.
Ooh.
So if you're going to tongue scrape, I think that's an old ancient practice that's been used for, hundreds, if not thousands of years in some cultures. Yeah, so if you tongue scrape, according to our research, you shouldn't use mouthwash, because when you tongue scrape and use mouthwash, that's when we see the highest increase in blood pressure in response to the mouthwash. But I think if you're just using tongue scraping once or twice a day without mouthwash, then our data suggests that that's better blood pressure control. And I think the analogy is you till the soil, right, before you plant your crops. I think what tongue scraping is doing is kind of tilling that soil in the crypts of the tongue and allowing, a more diverse microbiome and kind of building out the ecology of the microbiome.
Yeah,
So
no, I like that.
practice.
I think that's a great analogy. I mean, it gives me a great mental picture of what might be going on. So let's assume that somebody has an intact oral microbiome and they have a readily available dietary source of nitrites that they can convert into nitric oxide. Once we're in the body, what are things that can interfere with or derail nitric oxide production or prevent cellular responses? Because I know, I mean, technically nitric oxide has a receptor, which is it's an enzyme rather than like a receptor like we think of in terms of thyroid receptors.
Right.
So once we're in the body, what kind of stuff do we need to think about clinically that might be interfering with this?
Well, the first one that comes to mind is heavy metals. Heavy metals scavenge any nitric oxide that's produced or that may be produced by, dietary means. And in fact, that's the only people that we really realized over the past 10, 15 years that we couldn't normalize their blood pressure with nitric oxide or those with heavy metals. Things like mercury. If you have mercury amalgam fillings, mercury, you vaporize that with each, bite of food. Mercury, but things like cadmium, iron, cell-free iron, increased ferritin levels. Things like sickle cell anemia, that's responsible for the vaso-occlusive crises in sickle cell patients. You get this cell-free iron that then scavenges any nitric oxide,
right?
And it can't vasodilate. It causes platelets to hypercoagulate, and you get the vaso-occlusive crises in sickle cell patients. So that's really the main one. I mean, there's finite chemistry of nitric oxide. It reacts with cysteine thiols and redox active metals. So you've got to control the redox environment of the cell. And you have heavy metal burden. You've got to eliminate the heavy metals in order for nitric oxide to do its job.
Right. And I know that there's a handful of nutritional cofactors that need to be in place for this all to work. And we've mentioned THB or tetrahydrobiopterin a couple of times. BH4. My understanding of that is that's a folic acid derivative that's given to us by how our microbiota metabolize B9. And am I correct in that? Is there anything else I need to understand about that? And do we have polymorphism issues in BH4 production?
Well, in BH4 production, so there's a whole production cascade, and there's this redox kind of coupling system where MTHFR reduces biotin, tetrahydrobiopterin that feeds into the NOS pathway. So the MTHFR is what's responsible for maintaining this redox balance of BH4 to BH2. And that's kind of the redox ratio that determines whether NOS is coupled or uncoupled.
Okay.
So the MTHFR is very important in maintaining NOS coupling. But there are other ways that the body can get, tetrahydrobiopterin, get it through, methylfolates and intermediates. But there are other production pathways, endogenous production pathways. I think there's even a synthetic pathway that occurs through, the microbiome, both in the gut and probably in the oral cavity.
All right, so although I do seem to remember you saying at one point that the BH4, the role and the position that plays is that is the rate limiting step in NOS activity and nitric oxide production. So is that one of the reasons why do you think from a design standpoint that we have so many different ways that we can get it because it's so essential to the process?
Well, it's clear that the rate limiting step in NOS uncoupling and insufficient nitric oxide production by the eNOS and nNOS is through insufficient BH4 or increased oxidation of BH4 to biotin. So that actually prevents this disruption, the flow of electrons to oxidize arginine to nitric oxide.
Mm-hmm.
And in fact, that disruption in the flow of electrons reduces molecular oxygen to superoxide. So an uncoupled NOS actually generates superoxide instead of nitric oxide. And the primary way, there's many ways to disrupt that, but the rate limiting step in that whole process is the oxidation of tetrahydrobiopterin. So what we do, and what no one's been able to understand is, and you probably understand this because we're electrical beings, right? So there's what's called a redox potential, or a potential or a certain voltage that certain molecules need to extract or to steal an electron from another molecule. So there's an electrical potential that's required for that, and every molecule has its own redox potential. or a voltage that's required to steal that electron. And so if you maintain a redox couple at a certain electrical potential, then you can preserve the reduction of tetrahydrobiopterin and prevent its oxidation. And that's what we do that really nobody else understands. And that's kind of, you can only understand this if you understand the biochemistry and enzymology.
Right.
So, people have tried to copy our technology for years, but unless you understand redox potential and voltage within a cell and how to maintain that scavenging of electrons, then you're never going to get to where we are. We're light years ahead of any of the competition.
So what's the main driver of that redox potential, and is it different for the NOS system than it is for any other system?
So we've got a whole host of antioxidant defense systems. We've got water-soluble antioxidants, things like glutathione, vitamin C, cysteine thiols. Then we've got lipid-soluble antioxidants, things like vitamin E, other kind of lipid-soluble antioxidants. But and all these are redox coupled, right? So you can reduce oxidized glutathione to reduce glutathione by ascorbic acid. So these are redox coupled. And so that redox potential changes, kind of goes down this whole potential pathway.
Yeah.
So, and then we have antioxidant defense systems, right? Like thioredoxin, glutathione peroxidase, all these enzymatic antioxidant defense systems. So all of those require a different electrical potential in order to maintain or transfer electrons between molecules. And that's why we need a hierarchy of these, because every cellular system and every organelle within the cell maintains at a different kind of redox potential. So if you have an overwhelming oxidative stress, you're going to overwhelm these antioxidant defense systems, whether they're just kind of solution chemistry or they're enzymatic, like thioredoxin, glutathione peroxidase. So it scales the entire redox potential of what a cell would normally maintain. So you can interrupt different biomolecules at different stages of this, depending upon where the oxidative stress is occurring and which molecules it's oxidizing. Or if you've got some type of perturbation in your antioxidant defense system, if you're deficient in selenium, for example, then your thioredoxin enzyme is going to be compromised. And glutathione peroxidase, you need these metalloproteins and these trace minerals in order for these metalloproteins to work and function properly.
Right. So would it be the case then that, someone who's systemically inflamed and they have this increased oxidative stress, let's just characterize that by saying they've got low intracellular glutathione, for example. It's my understanding that oxidative stress is going to induce the INOS pathway, upregulate a lack of a better term, a crap ton of nitric oxide, which has the potential then to damage the local tissue where that's happening, because the nitric oxide produced through the INOS pathway is an immune strategy as part of the respiratory burst tactic. Poke a hole in something and kill it from the inside out. That's obviously non-scientific language, but that's my practical understanding of what's happening.
Well, let's go back to the reason why our body does that in the first place, right? It's part of the host defense mechanism. So the body really doesn't discriminate between a wound and an infection.
Yeah.
Right. So when we see that, we get an upregulation of what's called cytokines. So it's these cytokines that lead to the induction of the INOS enzyme.
Yeah.
And within 4 hours of a cytokine storm, you see an increase in expression of the INOS protein. Within 4 to 8 hours, you see maximal INOS enzymatic activity. So, we published on that back probably the early 2000s. So why does it do that? Well, there's either an injury to the body that it's hoping to repair and replace dysfunctional tissue, or there's an infection. So what the body does is it wants to wall off that infection, right? So we increase nitric oxide. If there's an infection, nitric oxide is antiviral and antibacterial. So it's going to kill anything that's at that site of injury or infection.
Yeah.
And then you're going to get the signs of inflammation, the redness, the warmness, the dolor, color, the signs of infection. And so it walls off that, and then it starts to mobilize stem cells to that site. So you can repair any injured tissue. You've done killed off the infection, and it's that acute inflammatory response that's responsible for cellular regeneration, keeping us free from pathogens. The problem is when it becomes chronic,
Yeah.
when you don't turn off the switch,
Yeah.
then you get this chronic overexpression. And that's where you start to get, host defense issues, autoimmune issues, and kind of the local chemistry that causes cellular damage.
So does the INOS pathway display, in essence, plasticity and self-perpetuation the same way something like NF-kappa B does?
Well, NF-kappa B is part of one of these cytokines and one of these pathways that's induced by the cytokines. And we published on this, I believe, in 2009. Part of what nitric oxide does is it downregulates NF-kappa B pathway, which just leads to this toxic damaging pathway. So there's multiple kind of signaling cascades that nitric oxide works on. But it is self-limiting because, now there's evidence that when you generate nitric oxide, there's a number of cysteine residues on these proteins, these NOS enzymes. So when you get a lot of nitric oxide being produced, you form these nitrosated species. It comes back and modifies the cysteine residues on these proteins and shuts down their enzymatic activity. So everything in the human body under normal conditions is self-regulated, right? Because you have to prevent the damage by the body's own actions.
Right. Yeah, but I mean, we see in many different pathways that auto-regulation can fail. So I guess the question is, I'm trying to figure out how to frame what's in my head. Do we get a runaway dysfunction where the shutoff switch doesn't happen, and so now we get runaway production of massive amounts of nitric oxide from the INOS pathway? And it's my understanding that because nitric oxide is nitric oxide is nitric oxide, prolonged tissue exposure to nitric oxide can potentially be detrimental and damaging, probably conditional upon other conditions or other factors.
Yep. Now you're thinking just like a lot of people were thinking in the late 90s.
Okay.
And the condition that they were considering was sepsis or septicemia.
Okay.
Right? Because when you get this chronic infection that gets into the bloodstream of humans, they die. Typically, septic patients die from end organ failure. The end organ failure is due to hypoperfusion. So you lose perfusion pressure to the brain or to the liver or the kidneys, and then they die. So the whole thought process was, is overproduction of nitric oxide in sepsis leading to the vasodilation, the hypoperfusion, and end organ failure in septic patients? A very rational kind of explanation. So in order to test this, they did a number of clinical trials where they gave a nitric oxide synthase inhibitor that was specific to the INOS enzyme. And they administered this, and it was a double-blind placebo-controlled study in septic patients. And what they found was that the nitric oxide synthase inhibitor killed more septic patients than the placebo.
Wow.
So that told us in the field that nitric oxide overproduction during sepsis isn't what's leading to end organ failure. It's the body's way of protecting in some manner, right? And so I think this gets back to, there's a lot of confusion in the field. And people think, oh, well, nitric oxide has been shown to be present in autoimmune disease. And so, yeah, if you read the literature, there's a lot of confusion. I have people come to me all the time and go, well, and if you read this paper, nitric oxide is shown to be damaging or pathological in this particular condition. And the example is like the police. When there's a crime, police always show up to the crime, or you hope they do, right? And so police are always at a crime scene. Does it mean the police caused the crime?
Right. Yeah.
No. Well, nitric oxide is the police. They're always going to be at a site of inflammation or infection or injury, but they're there to police the situation, try to maintain some control and order in these biological systems. But just because they're present don't necessarily mean they're contributing to the pathology of that disease.
Right. Okay. So because I think we can keep going down in more depth. Let me kind of switch gears a little bit and ask a couple of questions based on some things that we said earlier. We've talked about different SNPs that might be of importance, like the MTHFR pathway SNPs, BH4, eNOS, or just the nitric oxide synthase SNPs. Would you recommend from a clinical perspective, because we draw distinctions between gene carriers versus those who are phenotypically expressing, right? So I don't get bent out of shape if someone's got an MTHFR, like a homozygous profile, but their homocysteine is like 7. I don't really care about the SNP at that point. So is it a circumstance where maybe we should think about doing these SNP profiles, understanding that the report is the report, then there's the clinical reality, but at least we looked, and if the SNP profile is matching up with something that's happening in real life, that's when we can go, okay, well, there might be a genetic problem here, which then obligates us to go, we need some kind of an NO strategy, maybe for the rest of your life.
Right. Yeah, I'm not a big fan of these genetic maps because, like you, I think it's the clinical presentation. You can't blame your disease on genetics anymore, right?
Yeah.
Because we know how to turn genes to epigenetics.
Yeah.
So, I think if you're looking to kind of complete the puzzle, the clinical presentation, then I think you can do some genetic testing. But again, that's the blueprint for what the body can then build into, protein machinery, if you will. So it may predispose them for something, but it doesn't necessarily mean they're going to present clinically from that genetic SNP.
Right.
So I like that approach. It's more about the metabolic function of the patient than the kind of the genetic predisposition.
Yeah, I look at gene testing as kind of like I call it a tier 3 test. It's not my first thought. It's usually not my second. Yeah. And if we have to get to another line of diagnostics, that might be helpful there. In some cases. Another question that I have is, and I'm basing this question on something that you said. I watched a presentation you did to some luncheon group somewhere along the way. In this luncheon, you were talking about how endothelial-derived nitric oxide has hepatoprotective effects, and that nitric oxide produced in one area can go somewhere else, be metabolized by distant tissue, and then have an effect locally at that endpoint. So because this is ubiquitous, and because we're producing nitric oxide through the entire vascular system, we're producing it neuronally, intraneuronally, and obviously that tissue is everywhere as well. And then we have the inducible form being produced by the immune system. I mean, how do we sort this out? Like, I mean, have you guys done studies where you've tagged nitric oxide produced in one place and then tracked it to somewhere else? I mean, how does that work?
Yep. So I'll tell you how this originated. In 2007, we were the first to publish a paper showing that nitric oxide acted as a hormone. So before that, it was thought that the nitric oxide can only act as an autocrine or paracrine mediator, meaning a cell in which it's produced or a neighboring cell. But in 2007, we were the first to indicate that nitric oxide was a hormone. So similar to testosterone, whether you get a burst of testosterone produced by the testes or get a burst of nitric oxide or testosterone given as a shot intramuscularly.
Yep.
Then you get this spike of testosterone, and then that initiates a number of downstream signaling pathways. So what we discovered was that nitric oxide produced in the heart led to some hormone function in the liver that protected the liver from ischemia reperfusion, right? It would also do it in the brain, the heart, liver, every organ in the body. So that was my aha moment that, well, hell, all we got to do is generate nitric oxide in a single biological compartment. And then the mouth, the oral cavity, is the most accessible, non-invasive compartment we have. So I thought, well, all I got to do is figure out how do we generate nitric oxide in the oral cavity. And that was the genesis of a lozenge, based on our work in 2007, because I knew if I could generate a certain amount of nitric oxide over a certain period of time in the mouth, that it would protect the heart, it would protect the liver, the kidneys, the lungs, every organ, tissue, and cell in the body.
Yeah. And
that's what we've accomplished. Now, mechanistically, how's it doing it? We think it's doing it through binding of glutathione and transported as an S-nitrosoglutathione. It's back to nitrite. Nitrite is cardioprotective and vasoactive. It's modifying fatty acids. So you're getting these nitro fatty acids that, conduct nitric oxide activity and basically signal in a completely different mechanism.
I'm glad you stated that, because I had a question that if the half-life is less than a second, how does it get from one place to anywhere else without being used or degraded? So it seems to me that clinically, if we're doing some kind of a clinical assessment of nitric oxide status, however we might do that, it makes sense to do that in parallel with some investigation into their oxidative stress and their antioxidant capacity, whether that's looking at reduced versus oxidized levels of glutathione in serum or isoprostanes or anything like that. Like, do you have, from your knowledge bank, do you have any clue, like what should I as a clinician be looking for that I can actually test for that would give me clues to their oxidative stress levels?
Yeah, I think it's the major ones. Urinary isoprostanes are a good marker of oxidative stress. We look at things like sedimentation rates. These are more inflammation, but where there's inflammation, there's oxidative stress.
Yeah.
An increase in ferritin, obviously high sensitivity C-reactive protein. Those are the major markers. But in terms of glutathione, go back to the endocrine mediator of nitric oxide. It's binding to glutathione. But there's millimolar concentrations of glutathione, right? We only need nanomolar, femtomolar concentrations of nitric oxide. So 10 to the 6th more molar concentrations than we have nitric. So really, as long as you have nitric oxide being produced, there's always going to be sufficient glutathione around to then bind that nitric oxide and transport it and increase its half-life from 1 millisecond out to tens of minutes and sometimes hours.
Right. All right, guys, that is the end of that part of the podcast. Again, I apologize for the abrupt ending, but we were having some technical issues. Dr. Bryan and I are in communication, trying to find another date that we can finish off this conversation. And we're going to make it a little less biochemistry and try to translate this to a lot of clinical applications. So stay tuned. We'll be back with Dr. Nathan Bryan.